Today in this video class we are going to discuss about the levels of protein organization. Levels of protein organization. What are proteins? Protein polymers of L amino acids. Polymers of L amino acids. And there are 4 levels of structural organization for proteins. 4 levels of structural organization. They are primary, secondary, tertiary and quaternary. So proteins are polymers of alpha amino acids and there are 4 levels of structural organization. They are primary, secondary, tertiary and quaternary. Now we want to discuss these structures in detail. So first one is primary structure of protein. What are primary structure of protein? Primary structure means it is a linear sequence of amino acid. Primary structure of proteins are linear sequence of amino acids. Look into this structure. This is a primary structure of protein. Primary structure of protein. Here you can see 3 amino acids. 1, 2 and 3. Each contain R groups R1, R2 and R3. Based on these R groups, the amino acids may differ. So, each amino acid contains the amino group and the carboxyl group and the alkyl group. So, in the primary structure of proteins, these amino acids are arranged in a linear manner, that is linear sequence of amino acids. And these amino acids are linked by the peptide bond. CONH is the peptide bond. These are the peptide bond. So, the sequence of amino acids, they are joined by the peptide bond. A linear sequence of amino acids joined by the peptide bond. That is a primary structure of protein. Here you can see the N-terminal end. This is the N-terminal end and this is the C-terminal end. N-terminal is here and the C-terminal is here. One example for primary structure is insulin. Insulin. It is discovered by the scientist Frederick Sanger. Insulin. It is an example for the primary structure of protein and discovered by the scientist Frederick Sanger. It has 2 chain, A chain and B chain. Insulin has 2 chains, A chain and B chain. A chain starts with the glycine and B chain starts with the phenylalanine. And the A chain contains 21 amino acids and B chain contains 30 amino acids. Here you can see a disulfide linkage between the 7th amino acid of the A chain and 7th amino acid of the B chain. A disulfide link is present in between the 7th amino acid of the A chain and the 7th amino acid of the B chain. Similarly, another disulfide bond is present in the 20th amino acid of the A chain and 19th amino acid of the B chain. 20th amino acid of the A chain and 19th amino acid of the B chain. So, the insulin contains 2 chain, A chain and B chain. A chain contains 21 amino acid, B chain contains 30 amino acid and A chain starts with the glycine and B chain starts with the phenylalanine. Here you can see 2 disulfide bonds. One is between 7th amino acid of the chain A and 7th amino acid of the chain B. And next is 20th amino acid of the chain A and 19th amino acid of the chain B. So there are 2 disulfide bonds are present. So the insulin contains both covalent and a disulfide bond. So the primary structure depends on sequence of amino acids. or the number of amino acids in the sequence. The primary structure depends on sequence of amino acids or the number of amino acids in a sequence, and it contains covalent bond and disulfide bond. Covalent bond and disulfide bond. Example is insulin. So they are linear sequence of amino acid. Primary structure is the linear sequence of amino acid. Now coming to the secondary structure of protein. The primary structure describes the sequence of amino acids forming a peptide chain. But in the secondary structure, it refers the local arrangement of chain in space. So the secondary structure of protein gives an idea about the local arrangement of the chain in space. Local arrangement of the. These are some common secondary structures of protein. They are alpha helix, beta pleated sheet, loops, turns and bends. The common secondary structures of proteins are alpha helix and beta pleated sheet, and similarly the minor loops, turns and bends. These are the common secondary structures of proteins. Now coming to the alpha helix. The alpha helix is formed by the folding of the linear amino acid chain or the linear peptide chain. The linear sequence of amino acid is folded into a helical structure like this to form the alpha helix. It is right-handed. It is the most common and stable conformation. It is the most common stable conformation, and it is spiral in shape. Spiral. So this is the structure of alpha helix. The alpha helices are stabilized by the hydrogen bonds. Alpha helices are stabilized by hydrogen bonds, hydrophobic interactions, electrostatic forces and van der Waals forces. The alpha helix, it is a secondary structure. It is a common secondary structure, and it is most stable conformation, and they are stabilized by the hydrogen bonds, hydrophobic interactions, electrostatic forces and van der Waals forces. This is the structure of an alpha helix. Structure of an alpha helix. Here you can see the NHCO and is involved in the hydrogen bonds. That is the oxygen of the carbonyl group and the NH of the amino group. Carbonyl oxygen and amide nitrogen. Carbonyl oxygen and amide nitrogen form hydrogen bonds to stabilize the structure, and this bond is in between the first and fourth amino acid. The first and fourth amino acid. The hydrogen bond is formed between the first and fourth amino acid through the carbonyl oxygen and the amide nitrogen. Consider this as an amino acid chain or polypeptide chain. In this, the first amino acid and fourth form the hydrogen bonds. First and fourth forming the hydrogen bonds. So in alpha helix, the hydrogen bond is formed between the first and fourth amino acid, that is involved the carbonyl oxygen and the amide nitrogen. So by this linkage, by these hydrogen bonds, these alpha helices are stabilized. These alpha helices are stabilized by these hydrogen bonds. Now coming to the other secondary structure, beta pleated sheet. This is the structure of beta pleated sheet. They are extended lines. That is, they are extended in a zigzag manner. Extended in a zigzag manner. There are two types of beta pleated sheets. They are parallel and anti-parallel. Parallel beta pleated sheets and anti-parallel beta pleated sheets. Consider this structure. Here you can see the N terminal and here is the C terminal. The next sheet also, this is the N terminal and this is the C terminal. So this is a parallel beta pleated sheet. This is a parallel beta pleated sheet. Now consider this structure. This is another beta pleated sheet. Here this is the N terminal end and this is the C terminal end. This is in this direction. At the same time, in the second strand, this is the C terminal and this is the N terminal. So this is in this direction. So these are anti-parallel beta pleated sheets. Anti-parallel beta pleated sheets. And they are stabilized by hydrogen bonds. Hydrogen bonds. They are stabilized by the interchain hydrogen bonds. But in the case of alpha helix, they are stabilized by intrachain hydrogen bonds. But in this case, they are stabilized by the interchain hydrogen bonds. And the structural classification of these alpha and beta secondary structures are Pauling and Corey. Pauling and Corey classified these secondary structures as alpha and beta. And carbonic anhydrase is an enzyme that is an example for beta pleated sheet. Beta pleated sheets. Carbonic anhydrase is an example for beta pleated sheet. Some short peptides such as turns, loops, bends, etc. are involved in the joining of the secondary structures. Here like this, joining of the secondary structures. They are short peptides involved in the joining of the secondary structures. Turns, bends and loops are involved in the joining of the secondary structures. This is the parallel and anti-parallel beta sheets. Now coming to the tertiary structure of protein. The secondary structure is folded to form the tertiary structure of protein. The secondary structures are folded to form tertiary structure of proteins. That is the 3D arrangement of polypeptides in space. 3D arrangement of polypeptides in space. Then the tertiary structure also stabilized by the hydrogen bonds, disulfide bond, hydrophobic interaction and ionic bond. So they are the arrangement of polypeptide chain in 3D space. Most of the enzymes and myoglobin are examples of tertiary structure. Most of the enzymes and myoglobin are the examples of tertiary structure of protein.
Hey everyone, quick biochemistry basics here.
The tertiary structure of proteins are 3D arrangement in space. 3D arrangement in space. Tertiary structure is the functional unit. Tertiary structure is the functional unit of a protein. Functional unit. Now coming to the quaternary structure of protein. If two or more polypeptide chains are kept together, if two or more polypeptide chains are kept together, that type of structures are known as quaternary structure of proteins. For example, hemoglobin is an example for quaternary structure of protein. It contains 2 alpha chain and 2 beta chain. 2 alpha chain and 2 beta chain. And these quaternary structures are stabilized by the hydrogen bonds, hydrophobic interactions, van der Waals forces and electrostatic forces. They are stabilized by the hydrogen bonds, hydrophobic interactions, van der Waals forces and electrostatic interactions. So in conclusion, the primary structure is a linear sequence of amino acid, and the secondary structure is they are folded into alpha helix or beta pleated sheets. So this is primary, secondary, and this also secondary. Alpha primary structure, this is alpha helix secondary structure, and this is beta pleated sheet. Beta pleated sheet. And the folding of these secondary structures, alpha helix and beta pleated sheet, in 3D space gives the tertiary structure of the protein. This is alpha helix, beta pleated sheet and alpha helix. So this is a 3D structural arrangement, that is tertiary structure of protein. Similarly, if more than 2 polypeptide units, this is one polypeptide unit, this is another polypeptide unit, this is the other one, these are joined together, they are known as quaternary structure. The quaternary structures are made up of more than 2 polypeptide units. The example is hemoglobin. So these are the levels of organization of protein, 4 levels of organization of protein: primary, secondary and tertiary structure of proteins. So primary structure means it is a linear sequence of amino acids, and the secondary structures are alpha helix and beta pleated sheets. The linear sequence of amino acids are folded to form the secondary structures, alpha helix and beta pleated sheets. They are stabilized by the hydrogen bonds, and each turn of the alpha helix contains 3.6 amino acids. Each turn of the alpha helix contains 3.6 amino acids and stabilized by the hydrogen bonds. Then beta sheets, there are 2 types: parallel and anti-parallel. Parallel and anti-parallel. Then tertiary structure means the 3D arrangement of the polypeptide, and quaternary structure means the multiple polypeptide chains are arranged in a 3-dimensional space. So these are the levels of organization of protein. Thank you.